Hey, hi. Welcome everyone to Micro 461 Immunology. So my name is Howard Shen. I'm an instructor in our School of Life Sciences, and here is my information up on the screen. I always welcome you to email me if you have any questions about course content or any concerns about how you're doing in the class, or for any other reason. If you just want to chat and maybe seek some advice about your course of studies, or maybe ideas for different career paths, or if you find yourself very fascinated with immunology and want to just learn about more opportunities to broaden your understanding of the many different immunology topics that we'll cover this semester, just shoot me an email. In terms of office hours, I welcome you to send me an email and we can set up office hours either in person. You're welcome to come to the LSB, to my office, LSB 311, or we can have an appointment over Zoom. Okay, so I do it by appointment simply because from my experience, if I have standing office hours, most of the time it's just me sitting here without any students showing up, right, which isn't a great use of my time. So I think it's better to accommodate your schedule. If you want to talk with me, you just email me and I'd be really happy to meet with you because I always enjoy meeting my students and getting to know them, especially in classes that are bigger, right? On La Lima, you will see that there's the syllabus tool, and under the syllabus tool, there is the syllabus for this class that describes the grading scale, describes the different types of assignments that you're responsible for. Importantly, we are using IDAP, which is the interactive digital content provided by the publisher. And so on the Lima site, you will see links to the ebook. All right, so the textbook that we're using this semester is The Immune System by Peter Parham. This is the recent 5th edition. If you can only find the 4th edition, for example, if you don't want to buy this one from the UH bookstore, the 4th edition is okay also, although there might be some times when there's updated stuff in here that we'll talk about and you'll suddenly realize, oh, I don't have that. So you might want, at those times, want to maybe borrow a friend's book, photocopy it, whatever, if they have the newer edition. The ebook version is fine. In fact, the way it's set up is you have access to the current 5th edition ebook as part of your IDAP subscription, right? So you don't need to buy the textbook separately. Right. So you'll see the link to the ebook in there. It'll take you to the digital version of this textbook, and this is the book that we'll use for the course. In addition, there are, as part of the IDAP materials, these inquisitive homework assignments. And so for each chapter in the book, there are a set of questions. These are actually made by the publisher. And what I do is I go through the whole pool of questions and find the questions that I think are the most appropriate for what we cover in our class. So this is a good reminder. There's a lot of material in this textbook that we simply don't cover in our introduction to immunology class, right? Because this book is not written as, an intro to immunology for undergraduates, right? It's not. This just is immunology, and it's meant for people, sure, undergrads, but also graduate students, postdocs, or even physicians, immunologists who want to kind of review and make sure that they're sort of up to speed with their understanding of immunology. So there is a lot of content in this book that we just do not cover, because my approach to teaching immunology as part of this undergraduate course is I want to focus on the fundamentals that will help you really feel confident that you understand how the immune system works and all the various principles that underlie how our body, the cells, the tissues carry out their immune functions. Right? I would rather focus on a subset of topics and do a really good job making you guys familiar and competent with those topics, right? at the expense of maybe not covering some of the other stuff that's in the book here. So that does mean that the best way to study for this class, right, is to come to lectures, take good notes. Right? For every lecture, I give you the PDF form of these slides. You have them already. They're all in La Lima, so you can download them and sort of take notes on them as we talk. And then use the study guides. For every chapter in the book, I provide you a study guide. All right. Use those first, your lecture notes, the lecture PDFs, the study guide. And then, right, if you have the time, go through the book, but instead of reading each chapter from the start to the finish, right, skim through the chapter and find the stuff that we actually talk about in this class, right, and focus on those. That will save you a lot of time, effort, and headache, right? Because if you try to approach this class by reading the chapters first and then bail on lectures, right, because you're like lazy and don't want to go to lecture, but you're just going to read the book, you're not going to do well, right? Because you're going to spend a lot of energy banging your head on the wall trying to understand things in here that I don't even care about teaching you in this class, right? Right. So I'd rather you know what the important things are. And the way you know that is by coming to lecture, following the study guides, and then using the book as a supplemental source. And by the way, if you want a hard copy of the book, because this actually is a really good book. So for those of you who plan to go on and maybe pursue fields that have to do with human health sciences and immunology, like med school, for example, your IDAP subscription actually gives you a big discount on buying a hard copy of this, right? Because I think normally if you were just to buy this, it'd be like, well, I don't know, $150 or more, right? Typical overpriced textbook. But through your IDAP, I think you can get a copy of this for like $30 brand new or something like that. Okay, so that's something else to keep in mind. We'll talk more about how to use the IDAP features and the assignments as our semester gets underway. All right, but for now, I want you to think about this. Immunology, what is it? Immunology is the study of how the body copes with external or with exposure to external pathogenic, parasitic, or other perceived antigenic threats, as well as internal threats brought on by disease, right? So some of these terms I think you already know, right? So pathogens, you might have a passing kind of familiarity with what we mean by a pathogen, right? But antigens, antigenic threats, what's an antigen, right? I think you all have heard the term antigen, right? Now that we're 2 years or almost 2 years into the COVID pandemic, right? We now have a whole array of different sort of quick home rapid tests that you can take. You know, if you can find them in the drugstore, right? You can take these tests at home, and many of them are what we call rapid antigen tests, right? So what's an antigen? Right? We'll learn about that as we start learning about these different terms, okay? It probably doesn't surprise you that our immune system, a very important part of it, is to protect us against external threats, right? But maybe it's more of a surprise to think that our immune system is just as important for protecting us against internal threats, things that are going wrong that start from within our body, right? Like cancer, for example. Our immune system plays a very big role in that. One thing's for sure, this semester and probably the last 2 semesters of teaching immunology, right? The students who start in this class from day one, you're already a lot more sort of aware of thinking about your immune system and how it works, or maybe how it doesn't work, right? Because of constantly always being under this, COVID sort of umbrella, right? But one thing you might not think about that much is just how fundamentally important your immune system is for your everyday life, right? Not just COVID. So when you woke up this morning and it's the start of a new semester, you're probably anxious about your classes, right? Maybe you're happy that you're starting it online for the first couple of weeks, right? It's certainly convenient, right? You just roll out of bed, you're still in your pajamas, right? And maybe you are thinking about your immune system because you're thinking about, oh, it's good that I'm not on campus because I wouldn't want to be exposed, right? But typically, we don't really think of our immune system all that much. We usually only think about it when we get mad at it, when something goes wrong, right? So through the course of the semester, we will learn to appreciate how every single moment of every single day, your immune system is working, right? It's protecting you and it's keeping you alive. So the study of immunology then encompasses a wide range of topics, a big scope of different sort of knowledge that comes from the small molecular cell biology topics all the way up, right, to really broad topics having to do with the dynamics of diseases in human populations, how diseases like COVID spread, right, to form epidemics and global pandemics. So to start off with, let's look at this set of very nice photos here taken from the textbook. So these are different examples of pathogens. So we have different things like we have viruses. So like in this panel here, panel A is actually an image of, for example, SARS-CoV-2. Right? We have different types of things here like yeasts and fungus. We have bacteria down here. Maybe this is E. coli. We have pathogens that, like these squiggly things here, are about the size of cells in our body, like red blood cells. And there are pathogens, right, that are much larger. So for example, in panel I down here, right, this is maybe like a nematode, a worm, a parasitic creature, right, that can infect our tissues, right? And our immune system has to be able to protect against that as well. So here's an important definition to start off our semester. What is a pathogen? Right? Our definition of a pathogen is that it is a biological entity that has the potential to invade and colonize the human body, causing disease. Right? So this may be a single cell pathogen, it might be multicellular, or the pathogen might just even be a collection of biological material. So why do I say that? And why do I refer to it as a biological entity? Why don't I call it like an organism or something like that? Right? And that has to do with the fact that not all pathogens are technically living things. So there are four main categories of pathogens that all of our discussions this semester will focus on. And that is, for example, bacteria. So here's salmonella, like the salmonella that you're probably familiar with, that you might unfortunately get sick from in food poisoning, like undercooked poultry, eggs, things like that. Viruses, right? Viruses like in this picture here, hepatitis A, that can cause liver disease. Fungus, right, or fungi, such as the fungus that causes athlete's foot, right, that burning and itching between the toes of your between the toes on your feet. And parasites, multicellular organisms, such as a rat lungworm, right, which we're probably familiar with here in Hawaii because of things like unwashed lettuce that might have certain eggs of things like lungworm, that if you eat this sort of unclean, unwashed lettuce, you might unfortunately end up with these organisms in you. Right? So these are the four main categories of pathogens. And I wanted to point out that viruses, right, are technically not living things. Right? Viruses are little packets of genetic material with certain proteins, organized in such a way that these genetic material, the genetic instructions inside the virus, if they're allowed to enter a host cell, can hijack the host cell and force the host cell to carry out the instructions encoded for by the virus's genetic information. Right, so that's why technically viruses are not alive. Okay. And I know that oftentimes, especially with COVID and SARS-CoV-2, you say, yeah, but then how come we're always talking about, having things like, hand sanitizer to kill viruses, right? Well, in that sense, kill is used in sort of like a functional term, right? In the same way that, let's say, your cell phone, if it runs out of battery and you look down, right, and it has one bar left and it's red, right? You say, hey, my cell phone's dying or my cell phone's dead, right? But that doesn't mean you actually thought your cell phone was alive in the first place. It just means it ceased working, right? So likewise, when we say we're killing viruses, all we're saying is we're destroying the virus so that it cannot work to actually colonize and take over the cells of your body. So these four main categories of pathogens, bacteria, viruses, fungi, and parasites, they're all very different, right? When you look at them, you think, yeah, some of them are smaller than a typical human cell, like bacteria and viruses are, right? Some of them are larger, like fungus, and like this fungus is this branching type of structure. Some of them are really big, like a parasite like this lungworm is made up of thousands of cells. So what common trait do all pathogens have in common? I want you to pause and think about this for a moment, right? The common trait, I will tell you, is that all pathogens have a genome, right? There is genetic information carried by that pathogen that defines what that pathogen is. Right? The genes that are part of the genome of the pathogen, right, when they are expressed, when they are used, right, are critical to how that pathogen functions and how it's able to do something like infect the cells and tissues of our body. Right? So we are exposed to pathogens all around us every day in our daily routines. Right? I think the study of immunology is really interesting in the sense that it challenges us to think about how we're dealing with constant things around us that we can't even see. Right? Now, many of you are safely at home in your bedroom or at your kitchen table, right? And you're thinking, well, I'm not really surrounded by pathogens. Maybe some of you are in a Starbucks or something like that, right? But it looks pretty clean. But the truth is, we are constantly surrounded by pathogens in all of the different environments that we pass through in our daily life, right? In our home, maybe in our car or on the bus, maybe in the classroom, right? Etc. So when you look at these different scenes here, right, these different scenes of daily life, right, they look like they are all places that, yeah, we might encounter pathogens, right? These are different environments in which we are surrounded by pathogens, right? Even the surface of your desk or your table has pathogens on it, bacteria, virus particles, etc. But when you look across these different representative environments here, what environment is common to all of these different scenes or different scenarios? Take a look, right? What environment is the same in all of these? That environment is the human body. The human body itself is a self-contained environment. And in fact, the human body, the environment in our tissues, is the preferred environment for many pathogens. The bacteria that's in front of you on your desk or table right now, right, it doesn't necessarily, represent happy bacteria, happy to be sitting on a table. In fact, it's probably stressing out because it's sitting there, right, and it can't do anything. Right? It would much rather, for example, be in an environment where it can carry out its various functions, its various life cycle types of events. Right? So if we think of the human body as an environment, you suddenly realize every day we are a mobile environment that's moving through these different external environments. Right? And so therefore exposure to pathogens is just providing certain pathogens an opportunity to jump on board and get into the environment that they would prefer. Right? Now, I don't want to kind of freak you out here because I know some of you are probably like borderline germaphobes, right? And this class is probably not the best way for you to overcome your fear. But right before you drop this class, stick it out, because as we learn about our immune system, I think you will suddenly have a much deeper appreciation, right, for how our immune system is able to protect us against all of these types of challenges. All right. So the body is an ideal environment for many microbes. So in our gut, for example, our GI tract, right, there are over 1,000 different microbial species that just live in our gut. And in fact, the number of individual living microbe organisms that we carry around with us actually outnumber our own body cells 10 to 1. What does that mean? That means if you were to do something, here's a kind of a funny thought experiment, right? If I were to slowly take apart my own body, cell by cell by cell, and I started making two piles, right? I take a cell out, I'm like, oh, that's a human cell, I'll put it in this pile. And then I take the cell, oh, that's a microbe, I'll put it in this pile over here. By the time I've completely taken apart my entire body, right, there'd be 10 times more individual microbe cells in the microbe pile than there would be in the human cell pile, right? That's amazing. Now, I'm not saying that the microbe pile would be 10 times higher, because of course, all of these microbes, right, are teeny tiny, much smaller than a typical cell in my body. Right? But in sheer numbers, it's quite overwhelming. Just to put this in perspective, we carry around 5 pounds of bacteria in and on our bodies every day, right? So if you had a New Year's resolution and you're like, for 2022, I'm going to lose some pounds, right? Boy, it'd be great if you could shed 5 pounds of bacteria, right? That's kind of gross, right? But we carry that mainly in our GI tract, okay? But all these microbes, like the bacteria, right, that are in our GI tract, it's okay that they're there. They're not making you sick because these are considered commensal microbes. Commensal comes from, I think, Latin, which means to eat from the same table. So when you're eating food as part of your normal, daily routine, breakfast, lunch, dinner, snacks, whatever, obviously you do so that your body can derive nutrients from your food. But at the same time, all the good commensal bacteria that's living in your gut, they're happy too, right? Because they're sharing in the food that you're eating. So if commensal bacteria is good, right, then what's the difference between good bacteria and bad bacteria? Right? It turns out that good versus bad bugs are often just different strains of the same species of bacteria. For example, let's talk about E. coli, right? Now, when you hear E. coli and you think of E. coli in the context of the body, you usually automatically think bad, right? E. coli makes you sick. E. coli is bad, right? That's not necessarily true at all, right? For example, there's a strain of E. coli, so E. coli MG1655, which is a very normal, good commensal bacteria that's in your gut right now. Right? And this E. coli helps to break down the food that we eat, right? It does it because it wants to get the nutrients, but in the process of breaking down the food, it actually helps us digest the food. And so we get more of the nutrients from our food. This E. coli also, in the process of it carrying out its various life functions, it makes some certain vitamins that our body cannot make on its own, right? And these vitamins then get absorbed by our body. Right? And importantly, populations of good E. coli in your gut, right, compete against the bad bug populations, and they keep the bad E. coli strains, right, down so that they don't make you sick. So at the same time, there are E. colis that are bad. So this E. coli strain, this 0157H7, is a bad one, a really bad one, right? This E. coli secretes a certain toxin known as Shiga toxin. And this Shiga toxin, if it's absorbed by the cells of your body, for example, the cells that line your intestines, this toxin inhibits the ability of your cells to properly produce proteins. Right? So for those of you who have taken Bio 275 with me, right, the ability of cells to produce proteins is a fundamental part of a living cell. Right? And so if you actually halt the ability of a cell to properly make proteins, the cells will die. And so this leads to then bleeding of the intestinal wall, severe diarrhea, and in some cases, right, can actually lead to death. So this E. coli strain is the one that you may have heard about when you have things like breakouts of E. coli food poisoning in things like fast food chain restaurants, right? And yet it still is technically E. coli, just like the good E. coli that's in you right now, helping you. Right? So if this is hard to understand, think about it this way. Commensal organisms that are on our body or in our body, right, these microbes coexist in a balance. It's like this. Consider the family dog. Many of you may not realize that when you think about all the different types of domestic dog breeds that we as people enjoy, right, having as part of our families, there's only technically one species of domestic dog, and that's the Canis familiaris, right? It's just one single species, but many dog breeds, which you can think of as being like the dog version of a strain, exist. I mean, I think you guys know that different dog breeds can breed together, and that's how you get, different new breeds, right? So the fact that you can have two individual dogs that look very different mate and produce offspring, that right there tells you, yeah, right, they're the same species of animal. It's just different breeds. Okay, so you have a household where you have compatible breeds that might live in the same household in harmony, right? Like these four dogs here, right? Different breeds of dogs, but they're all happy dogs. They get together, they're pals, right? And they all share the same resources that are provided by the family, right? Food, shelter, water, love, attention, whatever, right? So the competition between these good commensal dogs, right, is in balance. But what happens if you add another dog to this mix, right? So the owners, they're dog lovers, they go to like a rescue and they bring home another dog, but oh boy, this dog has problems, right? This dog maybe is overly aggressive, right? And it starts fighting against the other dogs, out competing the other dogs for food and attention. This results in a very dramatic change in the household dynamics, right? Maybe even to the point where the owners realize, whoa, we made a mistake here. We can't keep this dog because he's messing things up too much. Right? So that's the same with the microbe populations in your gut as well. So consider this. In the colon, the large intestines, right, it's filled with commensal bacteria. And there's going to be, right, a few bad bacteria as well. But importantly, if things are in balance, then the good bacteria keeps the bad bacteria, right, from causing problems. So in this panel here, let's look at this cartoon. This is the wall of your colon. The wall of your colon, as with many of the mucosal membranes in our body that we'll learn about, is very thin and very delicate. In fact, it's made of essentially just one layer of epithelial cells. So this gray cell here, this rectangular cell, here's its nucleus, right, represents one cell in the wall of your GI tract. Right? Below it would be the tissues of your body that surround your intestine. Right? Above it, in this illustration, this empty space is known as the lumen. Right? So lumen is an anatomical term, and we'll use it over and over again this semester. So it's good that you learn it if you're not familiar with it already. Lumen simply means the space, right? The space inside of various organs in our body. For example, the space inside of our intestines, where all of our food travels, is the lumen of the intestines, right? And that's where you'll find all the commensal bacteria. So these purple bacteria here, we'll just say is good commensal bacteria, right? It's meant to be there. Let's say you have an infection, you get sick, and you take something like oral antibiotics, right? Like a pill, and these antibiotics kill bad bacteria. Right? The problem is most antibiotics don't discriminate between good versus bad bacteria. They just kill bacteria. So as a result, right, if you take a lot of antibiotics, you might actually end up killing a lot of the commensal bacteria in there. So its population goes down dramatically. Right? That's why many of you, if you've ever taken a course of oral antibiotics, It can do things like give you upset stomach and diarrhea, right? Why? Because you are actually eliminating lots of the good commensal bacteria that your body relies on to help you properly digest your food. But in this scenario here on this slide, we're talking about something even more serious. It could be then that it allows populations of bad bacteria to be able to then get a foothold, be able to then expand, divide, and take over. So this red bacteria is pathogenic. Maybe it's that bad strain of E. coli, right? And now it's able to outcompete the good bacteria. And what happens is secreting all kinds of nasty stuff, like those Shiga toxins that I mentioned. That's what these little red dots are. And so it's actually destroying the lining of your GI tract, right? And what happens when the cells of your GI tract, the walls begin to die off, right? Boy, you have a breach, right? You literally have a hole in your intestinal wall, and it becomes very leaky. And so this means things like blood and fluid from your tissues on the other underside begin to now seep out into the lumen. And that's how you can end up with, things like really awful, like blood in your stool, things like that, right? And importantly, now the bacteria that was normally in the lumen of your intestines now have a way to get into the underlying tissues. And the important thing about this is that not only will the bad bacteria have an opportunity to get through, but everything else that was in the lumen, including the purple good bacteria, they can also get through, right? And that's when you have big problems, right? Because now this bacteria is not just contained in the lumen of your GI tract where they belong. Now they're actually entering the tissues of your body. So what if we didn't have an immune system? So consider this. Right? Imagine a piece of old bread, right, that you kind of leave out. And in the air, there are fungal spores, right, floating around. We don't see them, but just like there's stuff on our desk in front of us, right, or even on our skin, right, there's stuff in the air. So if certain fungal spores land on a piece of bread, then the mold will begin to grow, right? So here is an illustration below of common bread mold, right? So this mold begins to grow and it carries out its genetic instructions to create different structures. Some of these structures, right, tap into the bread to kind of get nutrients from the bread. Some of these other structures are busy making spores to kind of spread. Right? So the spores are seeking a favorable environment in which to germinate, grow, and propagate. And so what does the bread supply? The bread is the host. Right? The bread provides nutrients, right, that these growing organisms need, the fungus needs. It provides moisture, so there's water in the bread. It's a favorable pH environment, not too acidic, not too basic. It provides structural support for the mold. Right? So if you look at the cartoon of the mold, you realize, yeah, in order for the mold to be able to form these structures, it has to have some supporting base to it. Right? And that's what the bread supplies. And the bread also provides some extent of physical protection for the mold. The very fragile structures of the mold, right, are supported by the bread. So the bread is the host, and the mold spores here in this example are what we think of as opportunistic invaders. It's not like the bread mold somewhere from across the room. It's not like the s'mores somehow sniff, but the spores across the room. It's not like they somehow sniffed out that there was a piece of bread left on the table and then they made their way over, right? No, they're just carried around by the air. And for every one spore that lands on the bread, just think how many spores don't land on the bread, right? They just land on the carpet or on the table. And too bad for them, they're not going to go anywhere, right? So the moldy bread then is just an unintended victim of this, because if you let the mold keep growing, eventually what happens, right? If you leave that moldy piece of bread there and you're just studying it, right, being like a geeky scientist type, right, you're like, don't throw it away, I want to study it, right? Maybe after a couple months you look at it and you're like, whoa, right, there's actually no more bread there, right? Every single bit of it is just mold, right, sort of in the shape of what once was the bread. And that's because the mold has used up all the resources, okay? But the important thing to keep in mind, again, is that microbes they don't have some kind of like malicious intent to harm their host, right? Because in fact, think about it this way, right? They don't benefit at all if they hurt the host, right? Because if a microbe kills the host, that is like the microbe shooting itself in the foot because they need the host to continue supplying resources. Microbes just simply carry out their genetic program of survival and reproduction as long as the host's resources support them. So damage to the host is simply just a consequence of, in this case, the mold spore carrying out mold spore functions. Right? But what about us? What about our bodies as this environment that's preferred as a host for many types of pathogens? Right? Our body's tissues represent many of the same resources, right? If you think about it, right? Nutrients, water, favorable pH conditions, structural support, protection, it's all there, right? So many microbes would be very happy colonizing our body's tissues. Right now, in the area around you, there's approximately about 5,000 mold spores per cubic meter of air. We typically breathe in something like about 2.1 cubic meters of air per hour. Therefore, in the time that you're sitting here during this lecture and breathing normally, there's going to be over 10,000 spores from the air that you will have breathed into your lungs, right? And that they're settling now on the surfaces of your lung tissues. Right? So just by the end of today's lecture, 10,000 of these spores have entered. Right? You better believe it's up to our immune system, right, to make sure that we don't become the next piece of moldy bread. Right? So this all starts, though, with physical barriers. We have physical barriers to keep pathogens out of our tissues. Right? So this is a really important concept for this class. All right, so it's really important that you kind of get this concept. What does it mean for something to be inside our body versus outside of our body? This is a very important distinction. Right? You know, ever since you're a little kid, you know, and you're sitting in your little high chair, right, and your mom or dad or whoever is trying to feed you some mashed up peas or something like that, right? You already had this concept of, oh, food goes in your mouth and then it's inside your body, right? And you swallow and it's in your body. I mean, hey, to this day, when you eat a sandwich, that's how you think about it, right? I'm eating this delicious sandwich and now it's inside of my body, right? But as immunologists, as budding immunologists this semester, I want you to think about this definition of inside versus outside differently. All right, because it's very important that we make this distinction. For example, when I was a little boy, whenever the car that I was in would drive through a tunnel in a mountain, I would always get excited, right? Because I'm like, I'm inside the tunnel, I'm inside the mountain, right? And I think, wow, I'm in this huge mountain, right? But when your car enters a tunnel, are you really inside the mountain? So for example, here's some photos of the polytunnels that I drive through every day on my way commuting to UH from Kailua. And then at the end of the day, I drive back again, right? As a kid, I was so excited. I'm in the mountain, right? But no, cars pass through the mountain, right? Through specific structures, the tunnel that's built to keep the car technically separate from all the material that makes up the mountain. Right? Cars pass through the tunnel without contacting the dirt and rocks that make up the mountain. Right? The concrete walls are a physical barrier. Right? So consider this. The car is not in the mountain, the car is passing through. Likewise, all of those commensal microbes in our gut that I talked about, like the good bacteria, right? They are not technically in your body. They have not penetrated the tissues of your body. They're kept in that special tunnel that is our GI tract, right? The walls of the intestines are like the concrete walls that keep the cars out of the mountain. Okay? So commensal microbes in our gut and other parts of our body likewise shouldn't enter the surrounding tissues. We have physical barriers too. And consider that in that example I showed you before, if you have a breach in the wall of your intestines and bacteria gets through, right, and actually gets into the underlying tissue, It doesn't matter if it started as good commensal bacteria or as bad bacteria, right? Because even the good commensal bacteria, once it gets through and it enters your tissues, it's not like the good bacteria is suddenly going to look around and be like, ooh, I'm not supposed to be here. I better go back out, right? No, right? The good bacteria will be like, this is great. I'm suddenly surrounded, right, by this new tissue environment that provides everything that I want. I'm going to go nuts and start multiplying. So the commensal bacteria becomes pathogenic bad bacteria if they break through and enter our bodies, right? And at that point, we would then consider ourselves infected. The moment pathogens pass through the barriers of our body that normally keep them out, and once they enter the tissues underneath, that is the start of infection. Okay? And we will explore sort of the technical issues of infection, of course, in subsequent lectures. All right, so here are some examples of physical barriers that separate, right, a body from the external environment. In blue are, for example, the skin, our external skin, our epidermis, which is a very obvious barrier, right? Our tough, impenetrable outer layer of our skin is multiple layers of dead, keratinized skin cells that provide an impenetrable barrier, right? Pathogens cannot get in. Right? But in red, you'll see that many of the surfaces that line different tracts in our body, for example, we already talked about the GI tract, right, our digestive system, but also our respiratory tract, or even our urogenital tract, right, that lead from the kidneys to our bladder or our reproductive organs. Right? These are all lined with barriers as well, but these barriers have to be quite different in composition than the tough, impenetrable layer outside of our body. But the key point of this slide is that, for example, the air that we breathe in into our lungs, right, is still technically outside of our body, even though it's filling up our lungs. Right? The urine in your bladder is technically already outside of your body, just waiting to flow down the ureter and to be expelled from your body. Right? And of course, microbes in your intestines are technically outside of your body, just as we said before. Right? So in all of these cases, right, there are barriers that are already defined that something is technically outside of our body, even though, you probably thought of food in your stomach or air in your lungs or the urine in your bladder as being inside of your body, right? Technically, they are not, right? Because they are just like the cars that are passing through a tunnel, through a mountain, right? So this is a very important concept for understanding infection, right? So make sure you're clear on that, what we mean by inside versus outside when we're talking in immunology speak, right? So when physical barriers are breached, for example, from cuts, scrapes, wounds, they allow entry of pathogens into the tissue under our skin. So touching, rubbing eyes, picking noses, your mouth also helps pathogens breach mucosa. Right? Breathing polluted air, eating contaminated food, or exposure to sick people all increase the risk of microbial entry. Right? I think that's pretty obvious. Right? So microbial entry into the internal part of our body, the tissues, is the start of infection. But the important thing I want you to keep in mind and realize is that the vast majority of infections, right, they're not something we worry about. We don't even realize it because they remain highly localized. Our immune system is really good at taking care of those infections right away. Within a few hours or days, they're fully extinguished, and you don't even realize that you experienced an infection. For example, when you brush your teeth or you floss your teeth, and sometimes maybe you accidentally jam your toothbrush too hard in your gums, or the floss between your teeth makes your gums bleed a little bit, guess what? You are experiencing an infection, right? Because if you've damaged the skin so that blood is coming out, that means all the microbes that are in your mouth can also get in. You are experiencing an infection technically, right? Because it's the very act of the microbes entering the tissue. That's the definition of infection. But, if you jam your toothbrush on your gum or you get a little blood from flossing, it's not like you're freaking out and worrying about it. You don't even think about it, right? Because your immune system takes care of that infection, right? And you don't even go on and think about it. You never feel ill or sick, right? Or any sorts of adverse effects. So this implies that we must have an on-call, ready-to-act way of detecting and neutralizing pathogens very quickly. And we do, right? Our bodies must be ready for diverse pathogens. We already said exposure to pathogens is a normal part of our daily life. We are literally surrounded by them every day, and they're getting into us all the time, right? We are experiencing multiple infections every single day of our life just by us doing our normal daily routine. So the vast majority of those infections are taken care of right away by our super effective immune system, right? Most of these pathogens are common everyday ones, right, that we've evolved to be able to recognize and take care of quickly. However, every so often, right, your body in the course of your life, you're going to encounter more exotic, uncommon ones that might be new to your body or new to the human species, right? For example, SARS-CoV-2, right, which is new not only to you, but it's new to the human species. Our immune system has to have evolved to be able to not only learn how to recognize those, right, but come up with defense mechanisms that you were not born with from day one, that you had to adapt, right, and learn how to be immune against. Right? So reacting to all pathogens equally would be a very inefficient use of our body's resources, right? Common everyday pathogens versus exotic ones. There must be different ways of dealing with them. Right? So how do we deal with familiar versus unexpected infections? Right? And it turns out our bodies must have two cooperating immune systems, a broad general response, right, and a more focused special response. And these are the two arms of our immune system, the innate and the adaptive. And immunology is the study about how these two work together. So hopefully this has sort of intrigued you into what we're going to learn about this semester. But I want to mention one more thing before we go, right? Because I know we're at the end of our lecture. I mentioned that maybe earlier this morning when you woke up and it's the start of the new semester, you're worried about how the classes are going to go, what they're going to be like, another online class semester starting off, hopefully just for a couple of weeks, and then we're back to in person. And maybe you thought about COVID and you're worried about COVID, right? But I want to sort of get you to think about this. Your immune system is working nonstop. If you woke up this morning and for some reason your immune system completely stopped working, right, and wasn't working and has not been working ever since you got up this morning, what would happen? Well, chances are you wouldn't notice anything at first, because the whole point of your immune system is it's working in the background, right? So you would not feel any different. In fact, you'd probably get through the whole day not feeling any different if you had zero functional immunity. But if you really woke up with a non-functional immune system this morning, then worrying about things like COVID or your classes and things like that would be the least of your concerns. Because if you didn't have an immune system starting from this morning, by this Friday, you would all be dead, okay? And not even from COVID, just from the things that you're being exposed to right now, right? So hopefully that's a scary thought, but it's also a fascinating thought. And hopefully it makes you want to learn more about what your immune system is doing right now to keep you alive. And that's what our course is about. And I look forward to having a really fun semester learning all this stuff with you. Okay. All right. So in the meantime, go ahead, make sure you check La Lima, get the syllabus and the schedule, and I will see you on Wednesday when we meet again. All right. All right. Bye-bye.